Hello and welcome to the next installment of a Bio 111 lecture. Lecture number 16 today is about Mendelian genetics. Like always, let's take a look at what we discussed last time. So last time I talked about variations in genes, and I said that changes in DNA, they lead to something that's called a mutation, a change. And these mutations can be detrimental, they can be disadvantageous, detrimental, or they can be advantageous, they can actually improve functions, or they can be neutral. And I said that variations in a gene are called alleles. And so a different version of a gene, same specific, it's the same gene, but a slightly different version of it, those are called alleles. And alleles can be detected by a number of biotechnological tools that we discussed. We talked about gel electrophoresis, very briefly about polymerase chain reaction, about a technique called variable number of tandem repeats, VNTR, and another one that's called RFLP. We talked about problems during meiosis and mitosis and how that can lead to having extra or missing chromosomes, and that state is called aneuploidy. We said that in humans, aneuploidy is often lethal, but some variants are viable, and we discussed some of those viable variants. And at the end, we talked about a phenomenon that's called polyploidy, which is the duplication of the entire genome and not just single chromosomes. Okay, so with that in mind, we are going to talk a little bit about inheritance today, inheritance of genetic material. So before we start, let's sort of talk a little bit about how these Mendelian rules were first found. Well, they're called Mendelian rules because they were found by Gregor Mendel. So these rules or these observations that he made basically bear his name. And while he must have been pretty smart and very diligent, he was also somewhat lucky in the choice of traits that he chose and the organism that he worked with, peas. And the reason is because he just happened to find genes for which all of his observations worked. Now, what do I mean with they worked? There are some generalities, and they work for many of the genes, but there are some exceptions, and we will talk about exceptions in the next lecture. But today we just want to talk about some general observations that Mendel made using peas. And the first important thing to recognize that he used in his analysis, in his experiments, was that he chose traits that were either dominant or recessive. And so here you have an example of all the traits that he looked at and that the two different versions that they came in. So basically what we have here is alleles, right? We talked about alleles earlier. Alleles are different versions of the same gene. And he happened to find alleles that were very, very defined and had exactly those two different versions that he was working with. So he looked at seed color, and the true alleles that he was working with were yellow and green, nothing in between. And the yellow one is dominant and the green one is recessive. He looked at the shape of seeds that was round and wrinkled. He looked at the color of the pod or the shape of the pod. He looked at the flower color. He looked at the position of the flowers along the stem or at the very end of the stem, flowers everywhere or just at the end, and the size of the plant. So we are just going to use, for the most part today, this particular trait here, this one trait that he looked at, and a little bit here also at the seed shape. So he picked traits, and that's really important, he picked traits that were clearly either dominant or recessive. And not all genes are that way, but he worked with those types of genes. Okay, so I want to take you back to the chromosomes because I find it so often easy to lose track of combining, the different types of information that we learn from the molecular to the organismal level. So when I say that a gene is dominant or recessive, really what that means for the chromosomal, on the chromosomal level, is that we have two chromosomes, right? They're homologous chromosomes. And in this case, you have duplicated homologous chromosomes with sister chromatids. And if we have two alleles, what that means is that the gene that sits somewhere on this chromosome for seed color sits on the maternal chromosome and it sits on the paternal chromosome. And it can be the same or it can be different. But in this case, in this example, what I have here is this particular gene here, denoted with this green line. That is the maternal seed color gene in the version green. So that would be the recessive version here. And here is a gene of the same gene on the paternal chromosome, and that is the version yellow. So here might be an organism that has these two chromosomes, and one of the chromosomes carries the allele for green, and the other one carries the allele for yellow. Now, if you have a trait that is dominant and recessive, that means that phenotypically, not genotypically, not what we find here on the chromosomes, but phenotypically, what we observe, one is dominant over the other. In other words, one is the one that you will see, and the other one will be hidden. So in this case, an organism that has this combination on their chromosomes would be phenotypically yellow, because that is the dominant trait. But genotypically, it would have one of each of the chromosomes. Okay, so that's going to be important later on for us to understand how alleles move and how they are given to the next generation. So I also want to just be sure that there's no confusion about the number of copies. So just remember from our discussion on the cell cycle that whether you draw a chromosome like this X-shaped with the two sister chromatids, or whether you draw it like this, it's the same chromosome, right? It's still, they're still homologs, and you would still have the maternal seed color gene here and the paternal seed color version over here. This is just a chromosome that has not been duplicated, so it would be after meiosis or mitosis and before the next synthesis phase, and this would be one that is just entering meiosis and mitosis after it's gone through the S phase, just so that you don't lose sight of that. Okay, so in a dominant recessive inheritance experiment, you usually, and Mendel did that as well, want to start with so-called true breeding parents. So true breeding parent is one that would always result in the same trait if you were just to look at the chromosomes from that particular parent. So in other words, and you'll see this later again, we're talking about a homozygous condition, not a heterozygous condition. So this, if this were the two chromosomes in one parent, this would not be true breeding, because it could give its offspring either this chromosome or this chromosome. Right? A true breeding organism would have only one or the other. So it would be homozygous for one or the other trait. This is a heterozygous state. Okay, so we would start with true breeding homozygous parents, and they could either have two chromosomes with a variant allele yellow or with a variant allele green. So these were then crossed, and in the next generation, you would look at the seeds and you would see that they are all yellow. All of these are yellow, and that is because of the dominance, that in the next generation you would only see one of the traits, the dominant one. So I say here, true breeding is that both parents have the same allele. And then when we have the segregation in the next generation, starting with the F1 generation seed, so this is a F1 seed from the previous generation, right? So the F1 plant, when you take that and you look at its offspring will now start to show the phenotype from the grandparents again. So in the F1, they all look the same, but in the next generation, the two phenotypes segregate again. So segregation in this case is a specific term in genetics that means the splitting of the dominant or recessive traits that are combined in the F1 into separate individuals in the F2. You will see that in the next couple of slides as well. So again, we're starting this particular figure here with the F1 seed, and we're looking at the F2 generation, and now we see that they're not all yellow. Some of them are yellow, some of them are green. Now, how does that happen? Why does that happen? Well, it happens at the cellular level because the chromosomes that are the alleles that are carrying the alleles are separated, right? So you have segregation during anaphase 1, and in such a way that one chromosome moves to one pole and one other chromosome moves to the other pole. And then in meiosis 2, these sister chromatids are split. That results in gametes that are either or, right? They're either one or the other. They're not both. And when they then randomly mate with another gamete, they pass down either one or the other trait. And that is why in the next generation, you could have two of the same kind, two different kinds, or two of the other same kind. So what I say here is that, well, how can a trait be segregated? How can it be sorted? It's because the trait resides on the chromosome. So the trait is here, this blue band, and it's one version, it's one allele. It's either the yellow or the green. And the chromosomes are then sorted into separate gametes throughout meiosis. Okay, so if you've taken any biology in high school, you've probably seen some of these slides before, some of these concepts. So what you have here is a depiction of this this genetic inheritance. So you start in the parental generation. So this is what we looked at previous, just in a different way. You know, it's just graphed or shown in a different way. So you had the plants that were true breeding, the parental ones, and in the next generation, they now all get one of the alleles from the father and one allele from the mother. And the way that this is often shown in genetics is by using letters to depict this dominance or recessiveness of a character. So there's some rules, and the rules for these letters are helping you later on when you're looking at something that's called a Punnett square. So a Punnett square, we'll go over the rules for the letters in just a second. But the Punnett square tells you how these letters or alleles, how they are sorted in the offspring. So this is our F1, and our F1 now is crossed with another F1. And if it's a plant, some animals do this as well, and not all plants actually, but P was just the right choice for Mendel, because these plants, if you just leave them alone, they will self. Now that means they have pollen and they have eggs, and the pollen contains the sperm. So if you self a plant, that just means, you know, you self-pollinate it, or the plant does it by itself, then you will have the same genotype in all the eggs and all the sperm. So that's convenient for the analysis. So if you want to draw this out and then see what the segregation pattern is, you can draw out this what's called a Punnett square. Punnett was a person, a geneticist, and so this is named in honor of Punnett. There's some rules that I'm suggesting that you follow when you are looking at genetic inheritance using a Punnett square. So if you're looking at one trait, so as we do here, seed color, yellow or green, then the Punnett square for the second generation, we're only ever looking at the second generation where we draw the Punnett square, not at the first, has 4 fields for 2 traits. So we have 2 traits, yellow or green, we have 4 fields. So then you ask, who are my two parents? So this is my first parent, this is my second parent. If this is a plant and it's selfing, then it might be the same individual, but it's still two parents genetically, because you have the egg and the sperm coming from the same individual, but nonetheless you have the formation of gametes through meiosis, and then the recombination of those two in the zygote. So that's why I'm saying here, note that selfing can occur in some species. During selfing, eggs and sperm come from the same individual. Okay, so now then you have to ask, what is the parent's genotype? In this case, the genotype is one allele, let's call it the big allele, from one parent, and the small allele, the little allele, from the other parent. And in this particular case, where we have a dominant and a recessive trait, we usually use a capital letter for the dominant and a small letter for the recessive trait. And then the way that the book colors this here is, it shows you the color of the seed. And since yellow is dominant over green, all the offspring in the F1 generation are yellow. And the phenotype, so the appearance, is yellow for all of them, because all of them will have this particular genotype. They can only have one A that comes, one large, one capital A, one big A from the yellow parent, and a little a from the green parent. And that gives you this mixed F1, this heterozygote F1. Okay, so once you know that you have a heterozygous F1, you now know the parent's genotype, and you write all the possible alleles on the left for the female. By convention, this is always the female, so it's the mother, and for the on the top here, for the father, for the male contribution. Now in this case, it will be the same because it's from the same plant, but one will be from this, one will be from that, will look the same. But remember that due to meiosis, the two chromosomes are split between gametes. So you do not have a big A and a little a in the same gamete, because they were split during meiosis, right? Remember how they were in anaphase, put into two different cells. And so therefore, this genotype would be producing gametes that either have the big A or the little a, and the chance is 50-50. So we'll put this here, and we put that here, and we put also this and that on the side, so that we know from one parent we're getting a big A and a little a, and from the other parent, this is the other parent, we also get a big A and a little a. And the book says here, expected ratio of big A versus big A versus little a is 1 to 2 to 1. That's the genotypes. These letter combinations, that's the genotypes. And so you can count this out, right? So you have the big A, where the big A comes from here and the big A comes from here. Or you have a big A, little a comes from here, big A comes from here. Here it's the other way around, with the big A coming from this parent and the little a coming from the other parent. And only in 1 out of 4 cases do you have a little a combination. And so that is the combination where you have two recessive alleles coming together. And at that point, the dominance is gone. There's nothing that's dominating over this green phenotype. And so now the phenotype shows up again as the recessive phenotype in the second generation, the F2 generation. So the expected ratio of dominant to recessive phenotypes, appearance, right, yellow versus green, is 3 to 1. And you can see that in this Punnett square. You can see it both for the phenotype, the colors, or for the genotype, the letters. Okay, so for the letters, the big A stands for the yellow version in this case. The little a stands for the green version in this case. The big letter always denotes the dominant trait, and the letter, whether it's big or small, stands for the same gene, right? So it's the same gene. We're looking at the same gene, not at different genes. It's the same gene with a different version, so different alleles. Okay, so if that makes sense to you, then that's already great, because that means that you hopefully understand how gametes split up their alleles due to separation of chromosomes during meiosis. Let me say that again. So during meiosis, you have the chromosomes and they split during anaphase, right? So there's one that comes from the mother, there's one that comes from the father, and that is what these two letters denote. One that originally came from the mother, that one that originally came from the father. They get split, and in the next generation, only one of them will make it into the meiotic product, so into the egg or the sperm. That's why there's only one letter here. There's two letters here, right? These are the gametes from this generation. So if you understand that meiosis produces haploid cells, not diploid, not both copies, just one copy, one version or the other, then you understand how they can then segregate in the next generation and combine in various possibilities, in various ways. So the big A here with the little a here, or the little a here with the little a here. We'll come back to that, of course. But I want to take a little detour because I always find it, personally I found it difficult when I was a student learning this, to make the connection between this view of genetics and the chromosomes and the molecular cellular functions that these genes have. And we've talked about cellular functions and molecular explanations for protein action. So I want to show you another way how you can think about dominance and recessiveness. So in this case of the yellow and the green peas that Mendel used, it turns out that in yellow seeds there's a gene that is this gene A that we call A in the previous slides, that produces this gene SGR. So there's a gene, it's the SGR gene. The gene is transcribed and translated. We talked about those mechanisms, and it produces a protein. And that protein now has a function. And the function of this particular SGR gene is that it helps to degrade chlorophyll in the seed. So the seeds are green because it's full of chlorophyll, right? So like a plant leaf, the seeds also have chlorophyll, and that's what makes the seeds green. Well, we have the yellow seeds and we have the green seeds. Why are the yellow seeds yellow? Well, they are yellow because the SGR protein helps degrade the chlorophyll as the seed matures. And so as the seed is maturing, the chlorophyll is degraded, and so therefore the SGR gene causes the yellow phenotype. Now, in the case of the green phenotype, the SGR gene has a mutation. That gene has a defect. That gene is still transcribed and still translated, but it makes a protein that is non-functional. So see how in my little drawing here, there's a piece cut off. So it's not complete, it doesn't function. So this protein cannot degrade the chlorophyll like the other version can. This allele can degrade it. This allele cannot degrade the chlorophyll. That means the chlorophyll stays intact. So if you want to now think back again at these letters of big A and little a, we can say that this is the allele version big A, and this is the allele version little a. The little a allele is not fully functional, and therefore the degradation of the chlorophyll does not happen, and therefore the seed stays green. While here, that allele, it's the same gene, but the gene is functional here, and so therefore the seed turns yellow because of that degradation that SGR is involved in. So now if you think about this from the two parents, whether it's the maternal or the paternal, and whether we are homozygous or heterozygous, big A, little a, that kind of approach, if we want to compare that approach of thinking, that way of thinking, here's an individual with two copies of big A, big A, big A. And so the maternal allele, the one that came from the mother originally, and the paternal allele that came from its parent, from its father, if they are both big A, then they both chromosomes produce the SGR protein, which leads to the degradation of chlorophyll. And it doesn't matter exactly where this protein comes from, it will degrade the chlorophyll in the seed. Now, if you have a heterozygous, big A, little a, you have an SGR protein that's functional. Let's say it comes from the mother. And then you have a SGR protein that is non-functional. And let's just say it comes from the father originally. So now the question is, well, if this is not functional, will the chlorophyll molecule stay intact? Will the seed be green, or will it be yellow, or will it be sort of in between? Well, this is what here's what dominance is. So dominance means, on the physiological level, that this protein that was made on one of the chromosomes, in our case here, the maternal one, that one is just also in the cell, right? So it's a protein that gets made from one chromosome, not from the other. The other chromosome is non-functional, but the one that is functional, there's enough of that there that will now act on the chlorophyll, and the chlorophyll molecule is degraded by the protein from the maternal allele. The paternal allele just sits there and does nothing, but it's enough for the maternal allele to be functional, and so therefore the maternal allele is dominant, right? So we see a dominant effect because whatever you look at in the seed, you will see the effect that the gene from the maternal chromosome has. That's what, molecularly speaking, dominance is in this case. Okay, so in a case where we have an individual with 2 copies of the little a, then we now have the case where both copies, the maternal and the paternal one, is non-functional. And so in both cases, you do not get the degradation and the chlorophyll molecule stays intact. So in this example, dominance works because one non-mutant functional copy of the SGR enzyme, this one, is enough to help degrade all chlorophyll in the seed. Doesn't matter what the other one is. Chlorophyll only persists if both SGR genes are mutant, from mother and from the father. And so that's, on molecular level, how the dominance works for the particular gene that Mendel worked with. He, of course, didn't know any of this. He just observed and turned out to make good observations. But now we know the molecular mechanisms for that as well. Okay, not all genes are doing exactly this kind of behavior, the dominant recessive behavior. There are some genes, a number of genes, that that show an intermediate phenotype. So here's an example of an intermediate phenotype. So this kind of intermediate genetics is called incomplete dominance. And you can see that in an example for snapdragon flowers. Here's a red one, here's a white one, and the F generation, the F1 generation, is pink. So if this was a dominant recessive trait, then let's say the red one was dominant, then you would expect the F1 to be completely red. Everything would be red. But that's not the case in this for this gene. So the two alleles are equal in their strength, you could say. And so therefore you get an incomplete phenotype, an incomplete dominant phenotype. It's a little bit red, but it's not really red. So in the next generation then, the Punnett square works pretty much the same way as it does for dominant recessive trait. You could write down the alleles that are possible, either the red allele or the white allele, for the mother on this side, for the father on this side. This is also a selfing plant, so you would have the same genotypes on both sides here for the parents, for the two parents. And then you could look and see, well, what would happen if you combined a gamete with a red allele with another gamete from the father with a red allele? Well, you would have a red flower. And if you had a red and white that are combined, you would have a pink one. And only in the case of two white alleles would you get a white flower. So there you would have a genotypic ratio of 1 to 2 to 1. Right? This one, there's only one of those, but there's two of these with the R and the W. And then the phenotypic ratio is the same as the genotypic ratio in this case. You have 1 red, you have 2 pink, and you have 1 white. So in incomplete dominant traits, mixing of alleles also leads to the mixing of the trait character. And the other thing that you might have noticed, is that the nomenclature is a little bit different here. So in order, you can't use the big A and the little a, or the C, little c, because that would suggest that the allele is either dominant or recessive, but it's neither here. And so therefore, C stands simply for color, and then you see this little superscript here for the one allele and the other allele, and they're both independent. Okay, so let's talk a little bit about probability of inheritance. There's a whole science about trying to calculate or try to determine the probability of inheritance of certain traits. If you go to a genetic counselor, for example, they will use probability in order to determine whether or not it's likely that the offspring of a couple that comes in for advice, whether their offspring might have a certain trait. Oftentimes that happens when there's a disease running in the family, and so then they want to know what's the likelihood that we pass this on to our children. So let's talk a little bit about how probability of inheritance works. So here's a Punnett square. Here's a trait. It's dominant and recessive. The big A is the dominant. The little a is the recessive trait. We have the two different types of gametes, here from the father, here from the mother. In the offspring, this would be our F2. These would be gametes from the F1 in a cross just like the one that we discussed earlier on. So we would have these different possibilities here for the outcome. So if you ask for the male gametes and the female gametes, what are the chances of inheriting a big A allele from the father? What are the chances for inheriting a big allele from the father? This is the father. These are the two alleles that he can produce. Well, the chances are 50-50, right? It's either the big A or it's the little a. So what are the chances of inheriting the big allele? 50%, or 1/2. And what are the chances of inheriting a big allele, or let's say a little allele from the father? Then that would be the other half, right? Also 50%. Now you can ask the same for the female, and you can say, well, what's the chances that you get a big A from the mother? 50%, 50% that you get a little a from the mother. It's 50-50 because the chromosomes are segregating during meiosis in one or the other way. Okay, so 50% chance, 1/2. So then if you were to ask, what is the probability that an individual will inherit a big A from the father and a big A from the mother? In other words, what's the probability that we get this phenotype? Well, the probability is 50% for this and 50% for that. And the way that you can calculate that is by following a rule, a genetic rule, that's called the multiplication rule. So the multiplication rule says, if you want to know something that can happen, but it would only happen this way, not any other way, then you can use the multiplication rule. So you would then multiply the 50% chance here, so 1/2 here, times the 50% chance here. 1/2 times 1/2 is 1/4, 0.25. So you could say that the probability of inheriting big A, that combination and only that combination is 1 in 4. And you can use the Punnett square to just check that, and you will see that 1, 2, 3, 4, there's only 1 out of 4 where that in fact happens. So then let's say, let's ask a different question. So if we were to ask, what is the probability that in this same cross, the offspring will be either big A, or big A, little a? In other words, what's the probability that the offspring of this cross will carry at least one of the big A alleles? Well, now you have multiple choices, right? You don't have just one choice. The big A was the only choice in the previous one where we used the multiplication rule. Here we have multiple choices. We have this as a possibility, we have this as a possibility, and this as a possibility. Now, what is the probability for getting this? Well, the probability for getting big A is 1 in 4. And the probability for getting big A little a is 50%. It's 1 in 2. So it's this one and this one. It doesn't matter whether it's big A little a or little a big A, right? It's just written this way because that's where the A comes from. So you would have a 50% chance to get these two, one of those two, and you have a 25% chance of getting that. And so the way that you can use the addition rule here is to add 1/4 plus 1/2, and that is 3/4. So in this little cross here, we can count this 1, 2, 3, right? Those are the 3 possibilities where the probability of having a big A, at least one big A, is given. So there's a 3/4 probability of this happening when you use the addition rule of 1/4 times 1/2, plus 1/2. Okay, your book has a much more complicated example. Let me walk you through this example. Try to calculate that out for yourself. See if the number comes out when you do it on your own calculator, and then see if you can add the multiplication and the addition rule together to answer this particular question that the book suggests as an example. So the question here is, what is the probability that any one seed out of 4 in this pod is green and that the others are yellow? So you see an example, any one seed out of the 4 is green, but the others are yellow. So it could be that this one is green and the others are yellow, or it could be that this one is green and the others are yellow, or the other combinations. So the probability that the seed, let's say start here, closest to the stem is green and that the others are yellow is determined by the multiplication rule. So there's a 1 in 1/4 chance that the seed is green, right? 1 in 4, little a. And there's a 3 in 4 chance that the seed is yellow. It could be either big A, or it could be big A, little a. So there's a 3/4 chance for this one, and for that one that it's yellow. Now, that whole combination, having these together, is this plus this plus this plus that. So that is 27/256. And if you wanted to know what the chances were that this seed is green in any of the 4 positions, you would then do the same thing for the other 3 remaining positions. So let's start with the second position. Well, it's the same thing here, right? 3/4 by 1/4 by 3/4 by 3/4. Same 27/256. And then there's the third and there's the fourth. And then you add that together. 27 plus 27 plus 27 plus 27 is 108/256. And if you divide that, you will see that it's 0.42, so that's 42%. The probability that any seed is green and the others are yellow is determined by first using the multiplication rule and then using the addition rule here to determine that the probability here. So I say here, to answer this, you use the multiplication rule first for each of the 4 possibilities, then you add those probabilities up because you're asking for the probability of any one of these 4 possible outcomes. That's why you're adding them. All right, so that's a way how you can use 2 rules that the book tells you about, the multiplication rule and the addition rule. And the main point of this is that you can use those rules to calculate probabilities of transmitting a particular allele to the next offspring, to the next generation. All right, so far we've looked at 1 trait at a time. Now let's look at what happens if you have 2 traits that you want to follow. So Mendel did that as well, and he looked at, again, at the trait with the color of the seeds, but he also looked at the phenotype of the shape of the seeds. So there were seeds that were either round or they were wrinkled. They had sort of a dried up, shriveled phenotype. So he set up this experiment here, and what that experiment is that he grew plants from true breeding strains that were yellow and wrinkled. So they had only those alleles. That's what true breeding means, right? So they did not have any of the other alleles. And the same thing for the green and round. So this was a plant from a true breeding strain that had green and round seeds. So he transferred pollen from one to the other. He looked at the offspring, and he found that in the first generation, all the seeds were yellow and round. So that means, since they were all yellow and round, that yellow was dominant over green. We already knew that from the previous experiments. And that round was dominant over wrinkled, because everything is round here, right? So they're all round, none of them are wrinkled. So it must be that the shape, roundness, is dominant over being wrinkled. And then he went to the next generation, and he found that in the next generation, segregation occurred, just like he found for the monohybrid cross, looking at only one trait. And if you look carefully here, you see that there are some wrinkled ones and some round ones and some green and some yellow ones. I'll blow this up in a second and you can see it better. So the expected ratio of these four types of seeds was 9 yellow, 3 green, 3 yellow, 9 yellow and round, 3 green and round, 3 yellow and wrinkled, and 1 green and wrinkled. That's our expectation now in hindsight. Mendel, of course, didn't have any expectations, and he just counted that. And he found these ratios. So he did this with hundreds and hundreds of plants and over several years, and he kept meticulous notes. And over time, he just did all the averaging and the analysis, and he said there's always a ratio of 9 to 3 to 3 to 1. There's always 9 that are yellow and round. There's only 1 that's green and wrinkled. How can that be explained? So, well, how that can be explained is in two ways, and I want to always bring back the chromosomes into this thinking, into your thinking, to help you think about this from multiple sides. So, if you have two chromosomes with two different traits that we are following, now we're following seed shape, and we're following seed color. They assume that they're on two chromosomes. That's one of the things that happened to be the way that they were on two chromosomes, and why I said at the beginning that Mendel was a little lucky with the traits that he chose. But look at how this can turn out to produce the certain kinds of gametes if we're now looking at two traits. So now we have the trait A, color, and the trait B, seed shape. During anaphase, our chromosomes are independently assorted. He came up with this term, and what that means is that they are either assorted like this, where you have the two big letters, the big A and the big B, moving to the pole, and the little letters moving to the other pole. So I say here, big A, big B on top, and little a, little b on the bottom. Or that the big A pairs with a little b, and the little a pairs with a big B. So independent assortment of genes in different chromosomes reflects the fact that non-homologous chromosomes can orient in either of the two ways that are equally likely. Bit of a mouthful. So non-homologous chromosomes are these two, right? They are not homologous chromosomes. Let's call them chromosome 1 and 2. These are chromosome 1 and chromosome 1. These are homologous chromosomes. Same genes, same orientation, potentially different alleles. Same thing here, but this is maybe chromosome 2 from the mother, from the father, chromosome 1 from the mother and the father. So these are homologs, these are non-homologous chromosomes. And they can orient, like we discussed before when we talked about meiosis, in two ways. They can either be like this, or they can be like that. And that means that they can give us 4 different types of gametes. They can either give us the gamete where big A and big B are together, big A and big B are together, or where little a and little b are together, yes, in this case. Or if the arrangement during anaphase was different, then we could get big A little b on top and little a big B on the bottom. So that results in these gametes for this choice of big A little b or little a big B. One cell can only do one or the other, right? But there are so many hundreds and hundreds of cells that are producing gametes that overall you'll get about half of this and half of that. That's why you have equal chance of getting this, or that in terms of allele combinations for your gametes. Let me say that again. You have so many different gametes that you're making, and not just making one. If you only made one, you would only get this or that. But if you make 100 gametes, then chances are 50 gametes will be like this, and 50 gametes will be like that. So you have an equal chance of getting this gamete, this one, or that combination. Okay, now let's go back to the Punnett square. We are talking about a dihybrid cross, right? We're looking at two different genes with two different allele forms each. And so I put this back here just to show you that now these gametes that I just showed you on the previous slide are the ones that you are putting into the Punnett square on the top and on the side. So on the side is the same as on the top. In this case of an F2 that is segregating from a cross that we had previously in the F1, where you had a big A little a big B, and a little b. So these are our combinations. Our combinations are now here. These are the possibilities that we can have in the F2. And if you then just look at the offspring and you just sort of draw it out, write it out, and then look at what that means in terms of phenotype, you see that you get 1, 2, 3, 4, 5, 6, 7, 8, 9 that are yellow and round. You get 3 that are yellow and wrinkled, these 3 combinations, 3 that are green and round, and only 1 that is wrinkled and green. And so this one, of course, is the one that is homozygous for both of the recessive alleles. That combination only comes out 1 in 16 times. So the ratio of the phenotypes is 9:3 to 3:1. Of the phenotypes, right? The ratio of the phenotypes, 9 yellow and round, 3 yellow and wrinkled, 3 round and green, 1 wrinkled and green. The genotype ratio is different. There's more than 4 different kinds. You can see that these two, they have a different genotype, right? They have a big A, big A, big B that will give you the same phenotype as if you had a heterozygous B allele, because the big B is dominant over the little b. Okay, so there is something that is called a test cross, and in lab we'll have you think about that in one of the questions. And so I want to talk about this a little bit here in lecture so that it's not completely new to you when you start working on that. A test cross is a cross that you do when you don't know the genotype of one of the parents and you would like to find out. And so why might we not know what the genotype is? Well, if you look at a seed and you see it's round and yellow, do you know the genotype? No, you don't necessarily, right? You don't know the genotype. It could be big A, little a, big B, or it could be big A, big B, little b, right? You don't know the genotype. You just know the phenotype. But if you wanted to know the genotype, how could you do it? Test cross. That's what you do. So a test cross, by definition, uses an unknown parent and crosses it with a homozygous recessive parent. So if I were to say, here's my parent, make a test cross, then you would say, this is my parent of unknown genotype, and it will be crossed to a known genotype because the test cross always uses a homozygous recessive parent. So the homozygous recessive would be the little a, little b parent. The offspring will then tell you the genotype of the unknown. I want to demonstrate that for you. So unknown genotype with known phenotype, let's say round and yellow peas. The possible genotypes for that round and yellow peas are big A, big A, big B, big B, this one. Big A, big B, little b, this one. And that's also the same as this one. And then the big A, big B, that's the same here and here. And then the big A, big B, little b, which you have here, 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 and here. So let's say we don't know what the genotype is. We do our cross, and here we do our cross, and let's assume we make this cross with our homozygous recessive, like you do in a test cross by definition. How would you analyze that? How would you now know? What would you have to do? Well, you could use a Punnett square. You could draw out a Punnett square. You would have to know. You'd have to do the experiment, and then you would have to count the offspring. You have to know what the offspring looks like phenotypically and what the ratio is of the phenotype. So let's see what we would do. In our example, we have an unknown genotype with a known phenotype. We cross it, and let's just say what would happen if this unknown phenotype was big A, big B, little b. We don't know that at this point, but let's just see what would happen theoretically if that was the case. So we crossed it with a little a, little b, and we make our Punnett square. Now our two parents are this one and that one. And what are the alleles? What are the gametes that this can produce? Well, it can produce. It has to have an A and a B in it, right? Because of the independent assortment. Everybody gets a chromosome 1 and a chromosome 2. It's not one or the other. You don't get two chromosome A or chromosome 1s or two chromosome 2s. You always get one of each chromosome. So these are non-homologous chromosomes. So therefore, one letter each. Okay, so the possibilities are big A, big B. Here's our possibility 1. Big A, little b, possibility 2. Little a, big B, possibility 3. And then the fourth possibility is little a, little b, and that's our possibility here. Now for this guy, what are our possibilities for the gametes? Well, it's little a, little b, or this little a and that little b, or this little a and this little b, or this little a and that little b. So this one doesn't make that many different types of alleles, of genotypes. All of the gametes will have the same genotype. That's the reason why you use a test cross, because it doesn't add any more confusion. It gives you exactly one type of gametes. And so if you write it like this in a test cross, you can, but you get the same results for each of these 4 columns. And that's why people oftentimes just fill in the first column or not even write out the other 12 squares. So we can now see in our test cross, if this was the genotype of our unknown, then we would get this kind of result. Right? We would get big A, big B, little b, and then these other combinations. And what are the phenotypes of these combinations? Well, you can think about this. We know that the big A is the dominant, so that's the dominant color. This is the dominant shape. So in the end, what we get is yellow and round, yellow and wrinkled, because now we have two recessive alleles. Here's the dominant one, so that's why it's yellow still, even though it's no longer homozygous, it's now heterozygous. But due to the dominance, this will still be yellow, this will be wrinkled. Here you have a wrinkled and a green, because now you are, in both cases, recessive. And here's the other way around, right? So here you are green, but you have the wrinkled phenotype, because that is the dominant one. So the ratio would be 1 to 1 to 1 to 1. And that would be the same here as well, right? This would give you exactly the same results as the first column. okay, our ratio would be 1 to 1 to 1 to 1 with these phenotypes if this was the genotype of our unknown. Well, let's look at a different genotype, an unknown genotype. Let's say our unknown genotype was big A, big B, big B. So then what would be the alleles that this genotype could produce? Well, they would be like this, right? They would be big A, big B, big A, big B, and that's the only thing that it can give us. Because this is not that much different from the test cross with a homozygous recessive, except that it's homozygous dominant. So it will only give us this kind. So in all cases, we will have a big A, big B, little b. The phenotype of the offspring would be all yellow and round. So how does that help us to determine the genotype of the unknown? If I keep saying, put something here, even though I say we don't know what it is. Well, the way that you do it in a test cross is you cross your unknown with a homozygous recessive, you look at the offspring, and then you look, what is the offspring? You have the phenotypic information if you look at the next generation. And then you try out every possible combination, just like we did here for 2. You determine the phenotype from the cross. So what do they actually look like? And then you compare to the expected for the possible parent phenotype. So if we had done this experiment and we got 400 seeds back, and all of these seeds were sorted, and we find that we have about 100 that are yellow and round, and 100 that are yellow and wrinkled, and 100 that are green and round, and 100 that are green and wrinkled, then this ratio fits. And so therefore, the probability that this is what the unknown is very high, because that's really the only way how we can get that kind of ratio of phenotypes in the next generation. So if you do this on paper, then you make a Punnett square. You just do a quarter of a Punnett square. You try the different combinations, and you see whether they come out with the expected segregation pattern from your experimental results that would be given to you. Okay, so let's spend a few more minutes on inheritance in humans. We'll spend quite a few more minutes later on, but not today. But I wanted to touch on how genetic inheritance is analyzed in humans. So Mendel used peas, and he used pure breeding individuals, and he crossed them together, and he said, well, what is the offspring? What does the offspring look like? Well, if you do that with human genetics, and you want to know what happens when certain individuals are crossed, that's questionable, right? So that's not really, for a variety of reasons, a experimental procedure that you can do. First of all, because it takes forever, because it takes so long until humans produce the next generation. And secondly, because we generally don't think that's okay to tell two people, who they should mate with. So therefore, human geneticists do it the other way around. They analyze what's called a pedigree sort of retrospectively. They say to a family, well, could you participate here? Could you tell us what the traits are? Could you tell me how you're related? And then I draw this all out, and then we can see how these traits are distributed throughout family members. And then from there, we can make some predictions as to how this trait is transmitted, whether it's dominant or whether it's recessive. There's some other ways, and we'll talk about these other ways as well later on in other lectures. So just some conventions here. When you draw such a genetic human pedigree or animal pedigree, you usually use a square for male and a circle for female. And then if there's a mating, so a couple, a couple that has children, and then you connect them with a horizontal line, and then they have their offspring, and all of these offspring here, is a large family, these two people had all of these offspring that have lines that come off of this long line, right? So all of the individuals that don't come off of this long line are mates of their children. So in other words, this couple had a son here, and that son got married to this woman. and so they have offspring, and that's their offspring. So if you look at just sort of in general how people are related, here you have a case of identical twins. That's just how this is written. Non-identical twins. We have this a double line that's a mating between relatives. So you see that these are cousins. Actually, these are brothers. No, sister and brother, and then their offspring are cousins. And so they're cousins, and their offspring is then denoted. That liaison is denoted with a double line, and their offspring comes from a mating between relatives. So that's how you draw a pedigree. And so you can draw a pedigree just by going through history, and you can ask people what they know, what they remember about a certain trait that their parents, their grandparents, their great-grandparents had. Or if you do this more recently, maybe there's medical information that you can use. So I want to show you two examples in humans that your book uses for looking at transmission of dominant and recessive traits in humans. So if you look at a pedigree like this, where you start with the fifth generation back, so these are the great-great-great-grandparents. That female here had a rare dominant trait, and this is the trait. It's called, what is it called? Polydactyly, something dactyly. So in this trait, the finger here is shorter, quite a bit shorter than it normally is, typically is. So if you look at a pedigree, and you want to know, is this trait dominant or is it recessive? You can do a number of things, and these call-outs here suggest things to look for, and I also summarize them up here. So if it's dominant, you already know that it's enough if one of the parents carries that trait, because that would be the big A, little a, right? So let's say this is the big A, or big A, little a, and then this person has little a, because you don't see the trait. Then in a dominant case, the single allele that carries the trait will be passed down to the next generation. So only one parent is sufficient to produce the next generation. You can see that here in all cases where the trait is inherited to the next generation. It's only one parent. One parent is enough, and you will get that trait. The other thing is that it's about 50% that are affected. So you see that if you count them, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 kids, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So out of those, 4 are affected, not exactly 50%, but thereabouts. So here it's a little bit more, here it's a bit less. If you sum them all up, you probably get to around 50%. You also see that males and females are both affected, and in equal numbers, relatively speaking. So overall, you have about just as many males as females. That's important because some traits are inherited on the sex chromosomes, on the X and Y chromosomes. We'll discuss that later in a later lecture, what happens when a trait is transmitted through a sex chromosome. So that is a good example for what a dominant trait looks like on a human pedigree. Now, if you look at a recessive pedigree, you'll see that, first of all, you see much fewer incidences of that trait. And another thing that you often see is that that trait is, it skips a generation, because if it's recessive, so that would be the green round P example, remember that from the F1, they were all yellow and round, right? So if that's the recessive trait that we're following, that would skip one generation, and then it would segregate in the next generation. And that's something that you see at first glance. Much fewer are colored in, are filled in, showing that particular trait that we're interested in. And then oftentimes you see that the trait shows up when there's a mating between relatives, because a rare trait is not that frequent in the population. And so if it stays within a family, then it can be, it's just more likely that it will then show up in the next generation when there's a mating between individuals from that same population, from that same family, in that same pedigree. And then another thing that shows you that this is a recessive trait is that often affected children have unaffected parents. So this person here has unaffected parents because they had maybe big A, little a, big A, little a. And so even though they were carrying that particular trait, the trait didn't show because it's recessive. But then when you happen to have little a combination, then that trait shows up. So when you have individuals showing the trait of parents who don't show the trait, that's also a good indication that this is a recessive trait in a human pedigree. All right, we'll talk more about human genetics in the next lecture. Let's just take a quick summary of what we discussed today. We said that some traits, but not all, are dominant or recessive. Dominant and recessive traits are caused by alleles carrying one of the two traits. We said that segregation of alleles happens because of the independent assortment of the chromosomes into gametes during meiosis. So in order to understand any of this inheritance, it's really best to understand meiosis. Mendel didn't know much about meiosis, but he understood what was going on with the transmission just by looking at these traits. That he didn't call them alleles, but at these traits that were segregated. So in retrospect, we can now much better understand that segregation happens because those alleles are independently assorted into different gametes during meiosis. We talked about a Punnett square. We said you can draw that out to get the probabilities of allele distributions, and a Punnett square allows the visual calculation of traits in the offspring generation. So we could just count out of those 4 squares. We could say 1 out of 4. We can say 1 to 2 to 1 is the genotype ratio, for example, or 3 to 1 might be the phenotypic ratio. And the same is true with the larger Punnett square with 16 or even 64 or 256 squares. It just becomes really cumbersome to draw that out if it's that many squares. You can calculate the allele distribution mathematically, and I spent a little bit of time talking to you about addition, the rules of addition and the rules of multiplication. We spent just a brief moment looking at incomplete dominance, which leads to intermediate phenotypes. I spent quite a bit of time showing you how test cross works, and I would encourage you to do this on paper yourself and just sort of see what would happen if you make the various combinations of the Punnett squares. But you will also do this in lab. And then we spent a little bit of time at the end talking about human inheritance and how you use pedigree analysis to study genetic traits in humans. And we'll leave it at that, and then we'll talk more about human genetics and passing down of traits through sex chromosomes and through other more complicated ways in the next lecture. I see you in class.